honorable keynote speaker of today's scientific seminar, respected doctors who are present here physically, and doctors who are attending through digital platform from different parts of the country. Good afternoon. Firstly, I would like to express my deepest gratitude to you for your contribution during this COVID-19 pandemic. You have been serving the humanity by risking your own lives every day. Big salute to you. Since the very beginning of COVID-19, SK+F is also privileged to serve the humanity standing beside our doctors community. Moreover, there was a clear vision from our managing director and CEO that SK+F will launch all possible treatment options to safeguard the people. I can clearly recall the early days of COVID-19. ওই সময়টাতে আমরা আপনাদের সাথে ডিসকাস করেছিলাম যে এই মুহূর্তে ঠিক কোন মেডিসিনটা আমাদের দেশে প্রয়োজন। আপনারা অনেকেই এবং বাংলাদেশ সোসাইটি অফ মেডিসিনের আর অনেক ডক্টররা বললেন রেমডিসিভিরের কথা। অতি দ্রুত এই রেমডিসিভির বাংলাদেশে আনার জন্য আমরা অল আউট অফ এফোর্ট দিলাম। অ্যান্ড দ্যাট ইজ হাউ it has been possible for SK+F to launch Remivir just within 7 days of its emergency use authorization by US FDA as world's first generic remdesivir brand. Similar case is MOLNUPIRAVIR. When the entire world was eagerly waiting for an oral anti-COVID, we were tracking all the clinical trials across the world. When we could see the promising outcome of MOLNUPIRAVIR in phase 1 and phase 2 trials, we started our preparation. একদিকে আমাদের ডেভেলপমেন্ট সায়েন্টিস্টরা এর ডেভেলপমেন্ট নিয়ে কাজ করে গেছে, অন্যদিকে আমরা অল ক্লিনিক্যাল ট্রায়ালসের আউটকাম বাংলাদেশ ডিজিডিএকে আপডেট করে গেছি। And that is how it has been possible for SK+F to launch Monuvir just within 4 days of its UK MHRA approval as world's first generic MOLNUPIRAVIR brand. You know, MOLNUPIRAVIR is authorized by UK MHRA, and you will be happy to know the facility from where SK+F is manufacturing it is also UK MHRA approved. So our people will get the world-class MOLNUPIRAVIR sitting here in Bangladesh. SK+F always believes in embracing technological advancements. For example, for treating cancer patients, our oncology facility is the one and only facility and one of the very few facilities in the entire world which is approved by EU GMP and ANVISA Brazil. We always strive to instill technological innovation in each of our products. For example, when people in our country are consuming esomeprazole in conventional tablet and capsule forms, SK+F took the initiative to launch the most updated multiple unit pellet system Isoral MAPS with tech transfer from renowned Evonik Germany for ensuring better efficacy and faster action. This is how we shall always remain steadfast in serving the people of this country with best quality medicine in all therapeutic classes, including COVID. Finally, I would like to pay my sincere respect to all the doctors who have sacrificed their lives during the darkest era of COVID pandemic. We will now watch an audio visual to pay our humble tribute to these great souls.
নেই শত্রু সেনার দল, তবু এ যেন যুদ্ধেরই বিভীষিকা। ঘরবন্দি ভয়ার্ত মানুষ, নির্জন শুনশান চারিদিক। কে দাঁড়াবে বুক চিতিয়ে মৃত্যুর সাথে যুদ্ধে? কোন সে বীর যোদ্ধা? মান মনে এখনো ছেলের অ্যাপ্রনের বোতাম সেলাই করেন, কিন্তু জানেন না জীবনের যে সুতো ছিঁড়ে গেছে তার কখনো জোড়া লাগবে না। অনাগত সন্তানটি হয়তো জানে না যে ভূমিষ্ঠ হবার পর সে আর কোনোদিন বাবা বলে কাউকে ডাকতে পারবে না। আদরের এই মেয়েটি এখনো বুঝতে শিখেনি বাবা যেখানে গেছেন সেখান থেকে কেউ কখনো ফিরে আসে না। করোনার মৃত্যুর মিছিলে ডাক্তার মেয়েকে হারিয়ে নিঃসঙ্গ বাবা আজ প্রায় বাকরুদ্ধ। মানবতার ক্রান্তি লগ্নে লড়াই করে বেঁচে থাকা নির্ভীক সৈনিকেরা আজ আবারও যুদ্ধে নেমেছেন হাজারো জীবন বাঁচানোর প্রত্যয়ে। তোমার হাত ধরে নতুন জীবনের আগমনে দেখি নতুন স্বপ্ন। তুমি দুঃসময়ে অসহায়ের চোখে সাহসের প্রতিচ্ছবি। তোমার জন্য প্রিয়জনের মুখে বারবার ফোটে সুস্থতার হাসি। তুমি আগামী প্রজন্মের চোখে মানবতার মহানায়ক। মানবতার এই ঘোর অমানিসায় নিজের ও পরিবারের জীবনকে তুচ্ছ করে চিকিৎসা সেবায় নিয়োজিত সকল ডাক্তারদের প্রতি SK+F পরিবারের গভীর শ্রদ্ধা।
Dear Doctor, now it's time for the most important part of today's program. I would like to call upon stage the keynote speaker of today's scientific seminar, renowned Professor of Medicine, the Line Director of NCDC DGHS. He is the Executive Member of Bangladesh Society of Medicine, who has been playing a crucial role in developing the national COVID guideline, and internationally acclaimed medical researcher, Professor Dr. Obed Amin sir, to please deliver his keynote presentation.
Honorable and respectable teachers, honorable and respectable directors from different institutes, respected physicians, doctors, postgraduate students, as well as the gentlemen, ladies, also the delegates from the home and abroad. First of all, thank you very much, the SK+F, for bringing me on this historic day of Bangladesh as well. I'm really delighted to have this opportunity created by the SK Pharmaceuticals to give a delivery on the part of Monuvir, which is one of the important historic revolution, I would say, that in the last 2 years. Gentlemen, ladies, we know the COVID-19 situation, the pandemic has been well controlled and stable in our country. But remember, the game is not over. If you follow the pandemic situations around the globe, surrounding the Bangladesh situation, even in the Southeast Asia region, as well as Europe and Western area, the situation is not very much stable. There is every chance that clusters are ever there, and there is still chances of developing the new variants, and which can create a problem in different countries as well. That includes the Bangladesh. So even though we are in a stable condition, the situations that can create a problem in the future means that scientific evolution and revolution must be carried on. And through this endeavor, I would be saying that it's a great opportunity for us to have our first oral anti-COVID drug in our hand. And so much so quickly that, again, I'd like to give thank you, SK+F, for giving this wonderful drug to our people of Republic of Bangladesh. So I will try to give something highlights on the part of MOLNUPIRAVIR, which is said to be the game changer in COVID-19. As I go through these many of the articles, I would like to say that we'll be giving this background information of the MOLNUPIRAVIR, how it develops. I will try to give some of the picture, how the mechanisms of the drug action happens in our body, which will be a little bit of tough. Thirdly, I will try to give how the trials has been going on in the animal studies, also the human studies in the phase 1, phase 2, and phase 3. Few of the interim analysis I'd like to share with you, and later on the recommendation that has been given, even though there is no still authorization, just an emergency use of authorization has been provided. So, gentlemen, ladies, with no more waiting, let's start. So, MOLNUPIRAVIR, the game changer in COVID-19. You see, this is the common scenario that has been situation in all around the globe. On the right side, it may be in Southeast Asia regional, low molecular income country hospital, or maybe on the left side you can see it may be a very advanced case of hospital in the United States of America. Wherever it is, the patients are worst, sometimes severe to critical, having a COVID-19, and some of them has a poor outcome in the form of the death. The issue is that science never rests when such situation happens. So you can see the search where the scientist is going on, and right now the concept is hit early, hit hard. That's the conception, because we did not find a magic things which can prevent the severity. Still, after these 2 years of huge amount of searching is going on, we're still not sure which of the patient is going to be in severe or critical, which of the patient can have spontaneous recovery. Although there has been some of these risk factors like the older age, maybe diabetes mellitus, hypertension, and even though there may be so much of beauty that risk factor can be biased by the physicians that have said it. But this drug, which has given some initial hope in the name of the remdesivir, and then came the big revolution in the name of the dexamethasone when it comes up as an immunosuppressive agent, because it has been found that this virus has an interim inflammation, and this needs to be stopped at some of the stage when the severity pulls back. And when this dexamethasone came up in the form of either tablet or injection, it revolutionized the treatment because it reduced the mortality, obvious in COVID-19. But since then, what happened? Let's see. So the current approval treatment for the COVID-19 as an antiviral issue was not very much used. You can see it was started from the remdesivir, which was injectable and IV infusion. And problem is that it was not consistently, and there was a variable responses. Some of the response was very good, especially if you follow this active trial in the United States of America. But uniformly, there was no constant presentations of the efficacy of this remdesivir all through the patients of the COVID-19. And that is the reason why WHO could not recommend it as a drug for choice. Then we have a tocilizumab. Initially, there was so much controversial issues about Actemra, but it was later proved that severe and critical illness, especially when the progressive hypoxia is present, the tocilizumab reduced the mortality. And last of all, this is the polygeneron. We all know that casirivimab and imdevimab. This combination gives a little bit of hope, because this is the agent which has been proven that it may reduce the severity of illness if we can provide it early. Big problem is that all of the agent that has been described is injectable form. It needs supervisions, you need to give to the hospital settings, and it needs expertise to give these drugs for any kind of amount. So this is a big problem, because the patient who remains in the milder illness or the moderate type of illness can remain in the home, and they will not go to the hospital settings even in the pandemic is going on. So how to resolve this? And the results came out in the form of this oral drug of MOLNUPIRAVIR. You can see the Medicine and Healthcare Products Regulatory Agency of the United Kingdom authorized this one, MOLNUPIRAVIR, for the treatment of mild to moderate COVID-19. Again, I repeat, this is the mild to moderate COVID-19. So this is the highlighting news. And also we find that it says that the Dr. Leon Retain is the MHRA chief executive. But the Molnupiravir is the world's first approved oral antiviral for COVID-19 and can be administered outside of the hospital setting, which is very important. It's an oral drug, we can use it even in the home. And then the chair of the Commission of the Human Medicine, Professor Sir Munir, you can see on the right side in his faces, with a big smile, said that he was found to be effective in reducing the risk of hospitalization or death in non-hospitalized adults with mild to moderate COVID-19 by 50%. So that was the issue why it becomes a big magic, as a name as the magic bullet. Well, Sajid Javid, it has been shown by you as well. The UHA authorization for these antivirals is an excellent addition to our MORI against the COVID-19 and will be also a game changer for the most vulnerable and immunosuppressed who will now receive a groundbreaking treatment. So that was the statement Sajid Javid given when the launching of this program happens as an emergency use authorization. All are good news, but let's start about the MOLNUPIRAVIR. So it looks like this. You can easily see this look like this structure. And it's orally available, but it's not a drug, it's a prodrug. And it has a nucleoside analog and hydroxycytidine. This is the main agents which is active within the body. The interesting point is that it exists in the two tautomeric forms, in the form of cytosine uracil nucleotide, and that exerts actually the antiviral effect. Now, the big issue is that when we are considering whether ribonucleic acid and other things, you know that there are 4 bases: adenine, cytosine, guanine, uracil. So I will be giving some of the picture how this drug really acts. Maybe it will be difficult to understand by all, but let me give a simple try. So these are the indications that actually provided. Right now we are giving it for the COVID-19 in the situations. But this is not the drug that has been given in the last 1 year, 2 months issue. It has been there in the pipeline since 2013. It was tried in the influenza, many other coronaviruses, not only the COVID-19 or SARS-CoV-2, but other seasonal coronavirus also has been tried by this MOLNUPIRAVIR. Encephalitis alpha virus in the form of Eastern, Western, and Venezuelan equid virus, and also the hepatitis C. So there are many attempts of this virus, antiviral drug, many of the viral agents. This has been initially used to be used for the influenza. Later on, the repurposing happened for the other viruses as well. So let's have some discovery fact. You can see the identification has been done in the form of the EIDD. EIDD means the Emory University, which initially started about the Institute of Drug Development 1931 as a replication. It was in 2013. In 2018, the MOLNUPIRAVIR actually overcome the bioavailability issue. In the December of the 2019, there was an emergence of the COVID-19, as you all know that in the China. In 2020 January, the Emory University, which is the Institute of Drug Development, planned the submission of the investigational new drug, MOLNUPIRAVIR, to treat the influenza. I told you this was started for the influenza. And even in 2016-17, when the Ebola gives a crisis around the globe, the MOLNUPIRAVIR was a trying effect. In the February, US FDA actually asked the Emory University to furnish all information about the MOLNUPIRAVIR against the pathogenic coronaviruses. In the March, the filing of the influenza investigational drug and licensing deal with the Ridgeback Therapeutics. This is the one small biotech company in the United States who initially came up with this one. In the April, the MOLNUPIRAVIR was licensed to the Ridgeback Therapeutics. In the May, Merck actually came up later on, acquired the exclusive worldwide right because they're a big company, and if you want to make drugs available all through the globe, we need to have a large company to come out. The Merck came up. In the August, you can see how quickly things are going on for the phase 1 clinical trial completed within the human. In the 2021, February, phase 2 clinical trial was completed. In the August, the phase 3 clinical trial started. And in this interim period of time, we already have few of the results of the phase 3 clinical trial. So you can see the approval came up at the November 2021. So within a very short period of time, the development was very crucial because we are very need to have a magic bullets. We do not have any magic bullets since this year's November came up. Before saying something about how molnupiravir works, I'd like to give a highlighting point to you how the virus actually causes replication in our body. We all know a little bit of it, but let's have some recap. We all know something about the spike protein, which actually combine with the ACE2 receptor. But let me tell you something, when they cannot combine, evacuation happens within the cell membrane. And after that, a trafficking happens with the incorporation of the cell surface of the virus. This surface is come from the cell surface of our body. After that, you can see there's a membrane fusion happens. Why this needed? This is needed because we want to release the virus particle to the cytoplasm. If we do not have the coating viruses, we will not able to release it to our cytoplasm. So you can see this is the genome of these SARS-CoV-2 viruses. But the big issue is that it needs to be replicated if you want to have a lot of viruses in our body and cell. So what will happen? It happens that the initial translation of this genomic virus happen and it creates lots of polyprotein. Look at this polyprotein, 1A and 1AB. So why these are needed? This is needed because we want to break this protein by a cleavage to have a large numbers of the non-structural protein. So you can see a lot of colors are there with the non-structural protein. And this actually is very crucial as because these are named as the RNA-dependent RNA polymerase. Or we can also say it's a replicase because we want to replicate the virus. We want a replicase enzyme. This is the enzyme. So you can see on the template of this virus genome, these non-structural protein, look at this one, 8, 10, 13, 14, there are 16 non-structural protein happen. And with this template, it actually causes the transcription to develop many of the important genes that is needed for the virus. So you can easily see virus is using our own machinery to develop its own viral particle. And this subgenomic known as the nucleocapsid, look at this one, spike, membrane, envelope. All the genomic tools has been done through the process where the non-structural protein is very crucial. And after that, what happened that the translation happened in our cell's endoplasmic reticulum. Virions then formation happened, combination, and then virion get a release after taking this cell membrane surface and go on. From one, you can have developed hundreds, thousands, millions, trillions, and so the devastation goes on and on. So this is the basic mechanism how the things happens. And I will give you a non-structural protein, huge number. I told you there are 16 non-structural proteins out there. These are very crucial part when we are doing the replication of the virus in our body. Now, when the replication happens in any viral genome, there's every chance that there will be mutation happens. And if the mutation happens, it has an intrinsic capacity to correct it. This is called the proofreading. And that is the reason why I told you the proofreading. So the proofreading causes the corrections of the genomic virus so that it can replicate of its own. Now let's come to the part of how molnupiravir works. We told you molnupiravir works on the part of RNA-dependent RNA polymerase. But so also by means of remdesivir, so also the favipiravir. So what's the difference then? So this is the difference that I'd like to tell you. Now when molnupiravir actually acts as a prodrug, it combines in our body and leading to the N-hydroxycytidine. This then combined through a kinase known as the NHC triphosphate or molnupiravir triphosphate. What I like to mention you that when molnupiravir triphosphate happens, it in the part of the genome just incorporate the M instead of the A. So you know the basic tool of the nucleotide is the A, G, T, and C. This is the nucleoside that is needed for developing the genome. All they have to do is just replace it through the M, T, P. So you can easily see that cytosine will be converted to uracil, and some of the adenine will be converted to G. So when this thing happens, the mutation will try to correct it by proofreading. But this is going to be happen again in the next cycle, again in the next cycle, and again and again. So you can easily see there are so many errors that happen in the virus genome. That virus could not correct it, cannot proofread this thing. They become hustle and bustle, and so that at every cycle generation mutagenesis increases and increases. and leading to the serious amount of catastrophe. So that is the reason why they call it error catastrophe. So what happened that through the mutagenesis two-step process, this molnupiravir not only causes the problem of the development of replication, but also inhibits the viral capability to correct the proofreading. That leads to the serious amount of virus and virus dies. This is how the mechanisms of unity for the molnupiravir. But what remdesivir has been doing is that they could not make so many errors. All they have to do is to make the chain termination early. So it cannot causes the problem of the mutation and causing the virus to hustle puzzle, just slow the rate of the chain termination. So that's the basic difference between the remdesivir and the molnupiravir. So you can see this two-step mutagenesis that I've been telling you. So in the first step, the enzyme synthesis negative stem actually is become problematic. And the second step, again the subgenomic RNA is synthesized by M-containing RNA, which is causing more and more mutagenesis responsible. So this is how it works. And this is the evidences of the papers that has been, I've told you about this molecular cell paper where the mechanisms, differences between the molnupiravir, the remdesivir, and as well as favipiravir is there. Although favipiravir has the capacity to go for some of the mutation and chain termination, the big problem with the favipiravir is that it has a very poor pharmacokinetic property. There are so many problems in the heart as well as the liver and kidneys. Remdesivir has also its limitation on the part of pharmacokinetics, as we all know. We cannot provide it to the part of the renal problems because it causes serious AKI sometimes. The big issue is that the molnupiravir has a wonderful pharmacokinetic property. There are less amount of side effects that I will show you in subsequent slides. So this is the paper that actually shows how the mechanism differs. This is another picture that I have taken up to show you wonderfully how it beautifully, this surprising G and M actually gets involved and replaces the normal cytidine of this virus messenger RNA. So you can see whenever there is incorporation M happens, it causes mutagenesis. On the right side, you can see the mutagenesis, which leads to huge amount of error, which the virus cannot correct. So let's come to the trials. I mean, when it is given to the animal, they choose one of the animal known as ferret. Now, when the ferret animal is chosen, because it can cause a mild to moderate illness, not to severity. It does not cause the lung involvement, because we want to choose those person who is having a mild to moderate illness, not the severe and critical. The problem with the mice and mouse is that whenever somebody given the virus inoculation, it quickly can transform into lung. So that's not our subject of interest. That is the reason why the ferret was chosen. And you can see these, this is the control group, and this is the group where the drug has been chosen to be given. Initially, through their nose, huge amount of virus loading has been done, and thereafter this drug is given as an oral pellet to check whether this drug can prevent the severity. And secondly, they want to also show whether the other contact animals get the infection or not. And thirdly, they want to see whether the drug is safe or not. So these are the things that has been shown in the printing. Look at this one. When the drug is provided, within 12 hours, the virus load reduces tremendously. It just came from the 10 to the power 6 to the 10 to the power 1 within the 12 to 24 hours. So that was the surprising thing that the compound concentration happened. But look at these concentrations in the control group. It remains over there, while the baseline has been touched within the 24-hour time. And it's been saying that almost all the virus RNA particles has been cleared up within 24 hours duration of time after giving these oral drugs. As because when the oral drug is given, the absorption was pretty quick, the pharmacokinetic property is very nice, the concentration jump up very quickly, and also the mechanism of action. So you can easily see this is the drug that is given this small dosage. This is another dosage, the red one. And they also see the responses. And you can see the response over here. You see the control has the 10 to the power 4 virus particle remains over there, but look at this one. When the drug is given, it just goes to the baseline. So that was the reason why the animal study was so much successful to be seen. And when the adversity has been shown, there was a very less adversity been found in this ferret. Not all of the ferrets actually dies. Even the control group, there was a severity, but there was no death. So let's get into the human trial, and it started with the phase 1. As you can see, to check whether the patient is safe or not, because phase 1 trial always believes safety first. And the side effect profile was most of them have a mild grade 1 reaction, and the percentage of the reactions to the mild, moderate, severe was same in the molnupiravir group as well as the placebo group. Even 1600 milligram was tried with a single bolus dose to check whether it is consistent with the phase 1 study of the human or not. And it came up good. So emergence adverse event was also check 1, and look at this number, 1, 1, and 2, and 3. Even the 300 milligram to 800 milligram was tried. And interestingly, severe adverse reaction, serious adverse reaction was found in 4 patients. Among them, one of the patients dies who was receiving the placebo. There was no death happens when the patient receiving the molnupiravir, even though patient have severe illness. This is another study of the phase 1 to see the safety of molnupiravir in patients with early SARS-CoV-2. Phase 1 open label dose skeleting. This study was based on whether how much doses we should be given, 200, 400, 800. And it came up that there was a tremendous benefit happens when the 800 milligram was chosen. And look at this one, side effect, cardiac, gastrointestinal, general, infectious, investigation, musculoskeletal, nervous system, remains 0, 1, 0, 0, 0, 0, everything. So you can see the organ-based serious adverse reaction was basically absent. An interesting pointer is that when 800 milligram was chosen, it was found to have good profiles on the side effect rather than 400 milligram, 300 milligram. And that is the reason why the dosage has been selected as 800 milligram. So this is the phase 2 study. And this has been published recently in the Matrix, which I have taken. 240 has the availability, and it was divided 200 milligram molnupiravir in 23 patients, 62 for 400, and 55 with 800, and placebo 62. So it's very stringent enrollment, allocation, follow-up, and analysis was done in this randomized control trial. What about the results? You can easily see visit was given in the day 1, day 3, and day 5 because the 5 days duration of treatment was provided. And when we're choosing about the placebo, the red number indicate the positivity of the viral in the day 5, day 3, and day 1. You see 200 milligram, 400 milligram, 800 milligram. Look at this 800 milligram. You can see there's a complete negativity in the day 5. Even in the day 3, it was only 1.9%. Look at the 400 milligrams virus particle who are also there. And look at this one. When we are considering about the dosage, drugage, and cumulative percentage of the responses, that was tremendously excellent when the 800 milligram molnupiravir was given. So the phase 2 trial actually shows not the safety issue, but the efficacy of a drug, of the molnupiravir in the human trial. About the adverse event, you can easily see the percentage positivity about the control and the different groups of the drug was remains same almost, 29% to 35%. It remains same, and there was a very less amount of adverse event. In the name of the dizziness, insomnia, some of the patients developed the diarrhea, and few of them developed the skin rashes. One of the patients has developed hives. That has been, I'm telling you. Let's come to the phase 3 trial. Although there has not been any published article over there, but the interim data as well as the other things has been came up, which actually shows that against the placebo, day 3 and day 5, the molnupiravir action, you can easily see that day 5, 0% COVID was positive at 200, 400, and 800 milligram. But it remains 224% positivity within this part of placebo. So what was the result actually? The result came up that 7.3% versus 14.1%. There was no death in the molnupiravir group, but in the placebo we found that there was 8 deaths happened. So at day 5, when the RT-PCR test was done, it was almost 0 when the molnupiravir was chosen, but it was around 24% when the COVID positive was there. But remember one important point that in this study, what is the remarkable thing is that when the RT-PCR was done, they have chosen also to see the genomic sequencing because we need to sure, is it the replicating virus still there or just an RT-PCR positive? Because we all know that RT-PCR does not necessarily means the infectivity. There may be viable virus and there may be non-viable virus. The good news is that this study actually shows whether the virus replicating virus or not. And it becomes 0. So that's the best part of this paper. So what was the extraordinary thing that has been shown? It's shown that it has 50% reduction of the hospitalization or death. So that's the magic that we are looking for. Is there any drug, if we given in the mild case, can reduce the hospitalization? Is there any drug, if we use in the mild to moderate case, can reduce the severity or the critical illness? It causes 50% reduction. That's wonderful. And there was no death in the study. At the same time, there was 8 deaths in the placebo control, we told you. Now, what I'm going to show you that has not been published, not been mentioned, few of the study done in the India. And in that Indian study, also the clinical parameters shown to be seen, as because we are all clinicians over here, most of them, we want to check whether the molnupiravir has any clinical improvement or not against the standard of care. It was 63.4% versus 22.3 on day 5, on day 79 versus 49. As the days goes on the second week, we did not get too much of the variations between the molnupiravir. So it has to be given early, because the tremendous benefit was found in the day 5, you can see. And not only for that, the faster time for the clinical improvement, molnupiravir versus standard of care shown that 8 days versus 12 days. Look at the p-value. Not only that, whether it can cause more RT-PCR negativity, yes, it is 77.4% on the day 5 versus 27.1%. And also the 94 versus 57 for day 10. So if you can provide it within the 5 days duration of time, there's every chance that you can reduce the RT-PCR negativity. It can improve the clinical improvement and also the faster time for the clinical improvement. Another study that has been in the phase 3 trial shows that the molnupiravir versus standard of care, 78.3% versus 48.4%. On the day 5, it becomes a big difference. But when it becomes day 14, look at this one, 99 versus 98. So you are not able to give this drug when the patient is severe. You will not able to give this drug when the patient taking a lot of oxygen. You cannot. Because there's no difference has been found between the placebo. So the selection of the patient is so much important. Otherwise, we will be in deep trouble. Faster clinical time also seen in the day 5, wonderful. It is around 80% versus 50%. Now, futility. What I like to mention that there was a few study done in the Indians in the phase 3 trial has the futility. Why? Because the patient was given a severe and hospitalized. There was a poor selection of the moderate to severe group. And those patients, when molnupiravir was given, have a more death. So that is the reason why you are going to be deteriorated patient if you select to give it in the severe to critical patient. Futility studies are there. And also there are so many ongoing studies. These are 5 of them, 10 of them. So this is going on. There's no interim results we have been found about this one, but the assessment is still going on. Hopefully we will get it in due time. So 4 facts about the molnupiravir is that there was a 0 death in the treatment group when the phase 3 is concerned. It has to be given within the 5 days of symptom. 10% patient treatment and placebo group reporting in the side effect only. 1% treatment and 3% the placebo group discontinued the drug because of the side effect. And the number needed to treatment is 15. Means that you need to treat 15 patients to avoid 1 hospitalization. You need to give 15 patient treatment by this mild to moderate case to reduce 1 death in the molnupiravir. Does that accept all of the variants? It does. You can see the gamma, delta, mu viral variants in clinical trials conducted globally more than 170 clinical trials, and it has been found to be effective. So how to take it? I think we have been discussing it. The 800 milligram is the ideal dose, so it has to be taken 200 milligram tablets, 4 plus 0 plus 4. Not only the mark, we also have Monuvir in the name of SK+F. Thank you very much again bringing this wonderful drug. Side effects, as I told you, sometimes you can have a dizziness, you can see, and sometimes people may have hive, but fortunately there was no fatality related to the side effects. Most of them are milder. Pregnancy, I'm afraid we are not going to give it any drug to this pregnancy because there was no study. Can we give it to the children or below 18? I'm afraid not, because we don't have any study. All the study that has been provided in the phase 1, 2, and 3 trial is given more than 80 years of age. So unfortunately, we are not doing. About the future plan, now the Mark has actually planned to give 10 million courses of treatment at the end of 2021. with more doses expected in 2022. We are expecting it. And the US has already agreed to deal for 1.7 million courses. So these are the huge number of crews that you can see in these online versions, those who are listening also seeing. And all of them are very much enthusiastic about the effects of the molnupiravir that has been shown in the different trial. It's a game changer. Does we have the magic bullets in hand? Can we use it? Can we use it prophylactically? So many questions are actually coming up hugely to this interim results of clinical items. Now, what I'm going to show you that has not been published is the meta-analysis that has been done on the molnupiravir. Even though we do not have a larger, huge number of trials, the amount of study that has been provided gives a meta-analysis. And you can see the Fisher, Hetero, Optimus, and Kuhn and Mark, all of them are randomized control trial. And about this one, we found that about thousands of the patients, enrollment and the control was 995. The 54% shows their clinical improvement. And among the all studies we consider, there's a 54% improvement was seen. All the lines were on the left side means that molnupiravir does have favors rather than the favor in the control. What about the meta-analysis of the improvement on the part of hospitalization? You can see there's a 57% improvement. About the study of Fisher and Hetero and all studies. So there's a serious amount of improvement of hospitalization prevention. What about the improvement in the part of early treatment? You can see the 89% improvement has been observed in the favors of molnupiravir. So if you can provide the treatment within the 5 days of symptom, remember, within the 5 days of symptom, then you can have an 89% improvement in this meta-analysis. So you can see the number of trials and Fishers and the improvement lists. I'm not going to exhaust you anymore. And it shows that green lines are actually more and more in the left side. And this is the baseline of the forest plot. So that gives it tremendously favors in benefit in the form of molnupiravir. So now let's what happened, Peter Horby, who is a professor of the University of Oxford, saying, he said that proportional reduction of the risk of hospitalization death is impressive. But it is important to remember that the absolute risk were 14% reduced to 7%. So quite a lot of people need to be treated to prevent one hospitalization or death. This means the drug needs to be very safe and affordable. The safety that also looks good. So again, this is encouraging. A virologist from the Vanderbilt University, the Nashville, Tennessee, we are very happy that one of the renowned molecular biologists from Vanderbilt University, Dr. Tanim Amin, is also with us. And he is also saying that this virologist said that I was pretty blown out by this one. Not in this year, he said it in the 2015. The ability of the virus have a so much unique that it can capable to say it's just blown away by any other drugs. And lastly, William Schaffner, he's the one who is the key person for the Moderna vaccine, you all know. He said if the government controls the distribution, then it may be more restricted, like the polygeneral and monoclonal antibody. But if left to the physician only, then it may be more widely, even in the beginning, it can be used. So monoclonal antibody are given via infusion or subcutaneous injection and require professionals actually to administer it. That's why oral molnupiravir has the potential to be a game changer. So the key message, gentlemen, ladies, number needed to treatment for molnupiravir is 15. So the drug needs to be very safe because we need to give 15 patient treatment to get one benefit. Molnupiravir has to be used early, less than 5 days of symptom onset. Again, I'm saying, do not use it in asymptomatic patient. Before someone is deemed ill enough when it don't work. Thirdly, the older age with at least one risk factor with mild to moderate illness at home is the ideal candidate to receive the molnupiravir. Next, lack of any study that is evaluating the role of molnupiravir in the breakthrough infection following vaccination, because it is going on all through the world. We need to have further study to establish whether it interferes with available vaccines or not. Cost effectiveness and accessibility for all is a major issue that need to be dealt with, and the effect of molnupiravir with other therapy for COVID-19 needs to also to be examined. So that's all. Any questions?
Thank you, sir, for your very enlightening presentation. I would like to request Professor Dr. Mohammad Robed Amin sir to please remain on stage while I invite Dr. Mohammad Murad Hossain, Senior Manager, Medical Affairs of SK Pharmaceuticals Limited on stage.
Thank you, Faiza. Bismillahirrahmanirrahim, and a very good afternoon. I am Dr. Mohammad Murad Hossain, Senior Manager, SK Pharmaceuticals Limited. First of all, I would like to congratulate and also like to give my thanks and gratitude to our today's brilliant speaker, my mentor, Professor Robed Amin sir. We are very much privileged to see a lot of senior teachers in front of us, but we are very sorry, time is very limited. Now we will start our question answer session, and question will be very limited, and we are very sorry that we are not able to collect all the questions from you, but I know you have a lot of questions and queries. So I request my volunteers to stand beside the doctors.
Can I ask?
Oh,
I have one very small question. The presentation was excellent. In your presentation, you have mentioned that RT-PCR assay and we can detect viable and non-viable. So is there any indication of the viral loads to start or to give the oral therapy with this molnupiravir? I mean, if I get the non-viable virus in RT-PCR test, and the patient is symptomatic, do we need to give this antiviral or not?
Thank you, Professor Tawhid. You see, when we are doing the test, RT-PCR positive, it actually cannot always reliably say whether we are doing some viable or non-viable virus or the replicating capable virus. So there was a few study done. One of the study done was the Professor Meena in the Harvard University. He shows that when we are seeing the CT value in RT-PCR, that can give us a good tool. So they said when the CT value is more and more, the virus load is less and less. Say the CT value is above 40, means that you are dealing hundreds of viruses. But if the CT value is less than 23, it indicates the billions of viruses. It may be trillions if it goes to the part of single digit. So that's the reason why they say that can we have a 15 to 38 within the range to indicate this is the replicating virus and the rest are not. So that's how somebody can say it's a replicating virus. But in this unique study, they did not choose it. They choose it to do for the genomic sequencing so that everyone can say, okay, it's a replicating virus. That's the best part that I'm like to say this. And if it is not a replicating virus, then we should not be watch or it. Why I'm saying this? Because when we are treating many patients and we are seeing that patient is completely normal, he can have a good life, but still then RT-PCR is positive on day 20, on the day 27, on the day 35, remains positive, and everybody becomes puzzled. I would say that do not be puzzled, check out the CT value. If it is more than 40, forget it, there's no replicating virus.
Thank you, sir. I know we have a lot of questions in our mind, but we are very sorry that we are not taking any questions. We will take the last questions because of time limitation. And lastly, I will request Professor M.A. Khan sir to please deliver your query.
Thank you. Thank you. I congratulate SK for bringing life-saving drug, and also thanks Robed Amin for a brilliant presentation. Nowadays, it is challenging to select the patient, ideal patient or correct patient for the drug. In our previous clinical experience, we found that about 30% patients have been negative RT-PCR, but clinically very much suggestive they are having coronavirus, that is both sides radiologically positive. In that case, we hospitalize and treat the patient. Now my question is that in that situation, if RT-PCR negative, clinically very suspected radiologically, as well as antigen test is positive, in some cases, can we start because the thing is life-saving? Thank you.
Absolutely, I agree with you, sir, because we are saying that's antigen positivity. So antigen positivity test, positivity remains an important issue whether it is confirmed or not. That depends on the prevalence. If any of the virus remains a high prevalence in a territory, then the antigen positivity can be given as a confirmed diagnosis. So if, what I would like to say that try to give a confirmation either by means of antigen test or RT-PCR test to make sure that you are in the correct path. Even sometimes if you have a negative because of the day of selection, which day are you using which test is very important for the COVID-19. In that case, the radiological evidence, any patient having loss of new onset of loss of taste or as well as smell or not. This is going to be a probable case of the COVID-19 by WHO definition. In that case, you can also select to give these drugs. But whenever you're choosing, try to choose it for the mild to moderate. That's the principle. And also, any patient having a single risk factor.
Thank you, sir. Thank you very much. And this is the ending of our question and answer session. Thank you all for your queries. We hope many of our concerns have been solved. This has been a wonderful afternoon. And to immortalize this event, I would like to call upon stage our Executive Director Marketing and Sales, our Executive General Manager Marketing, our Head of Marketing, our GM Sales, and our DGM Sales. Please come up on stage, and it is a great moment for us. We will provide a great moment for a great personality of Bangladesh, Professor Obed Amin, sir. Sir, I would like to request you to give the press to sir. Thank you, sir. Thank you all for your patience hearing. আমরা আসলে খুবই আনন্দিত যে আজকে আমাদের মাঝে আমরা সামনের সারিতে আমাদের অনেক সিনিয়র শিক্ষককে দেখতে পাচ্ছি। আমাদের পেছনে বসে আছেন বাংলাদেশের সকল নামি দামি স্পেশালিস্ট ডক্টরস। আমি সবার পক্ষ থেকে একটি অনুরোধ করতে চাই আমাদের সিনিয়র শিক্ষকদেরকে যে আমরা এখান থেকে 5 জন শিক্ষককে আমাদের আজকের এই সেমিনার নিয়ে, আমাদের আজকের এই প্রেজেন্টেশন নিয়ে, আমাদের আজকের এই মলনুপিরাভির নিয়ে কিছু বলার জন্য আমি শুরুতেই অনুরোধ করব বাংলাদেশের খ্যাতনামা গ্যাস্ট্রোএন্টারোলজিস্ট প্রফেসর ফারুক আহমেদ স্যারকে, ডিরেক্টর শেখ রাসেল ন্যাশনাল গ্যাস্ট্রোলিভার ইনস্টিটিউট অ্যান্ড হসপিটাল। স্যার, আপনি যদি আমাদের আজকের এই সামগ্রিক অনুষ্ঠান নিয়ে যদি কিছু বলেন।
Assalamu alaikum, respected dignitaries, respected participants. As a member of a team, as a leader of a COVID fighters in a dedicated COVID hospital, like you, I also myself observed over the last couple of years that how the science, how the scientific people was fighting incessantly, continuously to bring up a molecule, to bring up a armamentarium to fight this deadly COVID infections. And with good hope, we could find that, yes, the science and the scientists could be able to come up with arms that could fight against COVID virus. And right at this moment, we could be able to see light at the end of tunnel, that now we are getting more and more molecules, more and more drugs to fight this infection. And definitely this molnupiravir is one of these. And this is a real good thing for us that we are getting a good molecule, and hopefully in near future we will find a real magic bullet to fight this COVID infection without any failure. We are hoping for this, and I must give my heartfelt thanks to Professor Obed Amin for his impressive and high quality speech on this molecule, and I must also give my heartfelt thanks to SK for bringing up this molecule in our market, local market, much earlier than many other countries in the world. So this is a good news for us, good news for our pharmaceuticals company and the pharmaceutical market. I want to request one thing to our pharma market, that you are doing a lot of things to bring up the molecules in the market. Now I think we should move towards the process to go for research to find out a new molecule by yourself, not only to purchase new molecule from other companies. You have to bring up new molecule by yourself. So you have to go for research in this field, and that will really serve our purpose absolutely. Thank you, everybody. Thank you.
Thank you, sir. Thank you for your nice words. Now I would like to request Professor Shopon Bantopadhyay, Head of the Department of Radiotherapy, Dhaka Medical College. Sir, please.
Thank you very much. Before coming to this dais, I was remembering the best person of entrepreneurship, Mr. Latifur Rahman, the founder of Transcom and the owner of this SK Pharmaceuticals. Because some of the dream personalities of the country, they do their best job. You see that just 5 or 6 days back, the molecule was approved in the UK market. And just 5 days later, it is over here in Bangladesh. So big salutations to the group of Transcom and Escape. Secondly, it is Professor Robed Amin, the very dynamic personality. Just I must congratulate you as a brilliant speaker and to bring the whole coverage of this molecule, pros, cons, and with all statistical support so that we might be, the clinicians might be convinced with this molecule. One moment we must be keeping in our minds, that is we are very abusive in the prescription manner. When we do get some oral drugs, we just write it down, never seeing whether it is RT-PCR positive or antigen positive, whether it is prescribed for 5 days or 10 days. Please do be rational when you are prescribing such type of molecular drugs. And one of the moments to be taken care of by the DGHs, especially I do request Professor Obedanami as well as his team to put his surveillance under the coverage of DGHs to understand the post-marketing study of this brilliant molecules, because before saying a magic bullet, we need lot of ways to overcome. And finally, it is Her Excellency Pradhan Mantri Sheikh Hasina who played an enormous role throughout the world, especially by bringing vaccination earlier in this country. And I think her leadership as well as the entrepreneurship of this beautiful organization like Escape brought the molecule within 4 days after marketing approval in the UK. Thank you so much. We'll be fighting together against COVID. This is a brilliant evening. I think we'll be starting prescribing judiciously. Thank you so much.
Thank you, sir, for your nice words. Now I would like to request Professor Mohammad Nozul Islam, sir, Professor of Rheumatology, Bangabandhu Sheikh Medical University. Please, sir, have your speech, please.
আসসালামু আলাইকুম, ধন্যবাদ এসকেপকে এমন একটি সুন্দর অনুষ্ঠানের আয়োজন করার জন্য এবং আমাকে একটু আপনাদের সামনে দু একটা কথা বলার সুযোগ দেওয়ার জন্য। বিলভড রোবেদকে ধন্যবাদ। সায়েন্সের একটা সুন্দর রিফ্লেকশন আমি আজকে দেখলাম। আমি যদি পুরো সময়টুকু যে এনজয় করেছি সেখানে খুব পরিষ্কার করে বুঝে গিয়েছে রোবেদ যে কথাগুলি খুব সুন্দর করে মনে করে দিয়েছে ক্লিনিক্যাল প্র্যাকটিসে অত্যন্ত জরুরি। আমার রিমাটোলজিতে আপনারা জানেন নিজেও আমি কোভিডে সাফার করেছি। ফ্ল্যাভিপিরাভিরের সেই অনেকগুলি ট্যাবলেটও খেয়েছি। ফর্চুনেটলি হয়তোবা জটিল কোনো সমস্যায় ভুগি নাই, আবার আপনাদের মাঝে ফিরে এসেছি। আমার অনুরোধ থাকবে যে আমাদের একটা ক্রুশিয়াল জায়গা আছে এই জায়গাতে, সেটি হলো আমরা সাসপেক্টেড কোভিড। আমরা যখন আমাদের মানুষগুলি মারা যাচ্ছিল তখন আমরা দেখলাম মোর দ্যান 50% কেস দ্য সাসপেক্টেড কোভিড। এখনও যে পরিস্থিতিটা রয়ে গেছে, রোগীদের কাছে অনুরোধ যাতে এই প্রক্রিয়াটা সুন্দর করে হয়। আমরা আসলে অ্যান্টিজেন টেস্টটা সবসময় করাও যাচ্ছে না। বাইরে যখন একটা কোভিড পেশেন্টের আমরা সরকারি প্রতিষ্ঠান ছাড়া টেস্ট দিচ্ছি, 2500-3000 টাকা এখনও ব্যয় হচ্ছে। ইটস এ বিগ প্রবলেম। আমার সাবজেক্টে আমরা একটা সার্ভে করেছি, সেই সার্ভেতে আমাদের প্রেভেলেন্স ছিল 7.78%। ওই সময় আমার শুরুতে, সেই সময় অনলাইনে আমার এই কাজটা করা, এবং ওই সময় ইটালিতে যে সার্ভে হয়েছে ওনাদের প্রেভেলেন্স ছিল 8% ইন রিমেটিক পেশেন্টস। এই তথ্যটা আমার নতুন করে আমরা একটা কাজ করছি। আমরা এখন দেখছি কোভিডের প্রেভেলেন্স রিমেটিক পেশেন্টে কেমন, এটি হয়তো আমরা আপনাদেরকে কিছুদিন পরে জানাতে পারবো কত পার্সেন্ট। যদি এমনই হয় 7-8 বা 9-10% রিমেটিক পেশেন্টস কোভিডে সাফার করেন, তাহলে এর মাঝে অবশ্যই একটা সার্টেন গ্রুপ অর্থাৎ 4-5% মাইল্ড মডারেট এরকম তীব্রতা নিয়ে আসবেন আমাদের কাছে। আমরা যারা এই রিমেটিক পেশেন্টগুলিকে ডিল করছি, ট্রিট করছি, আমাদের আসলে এটা দেখা দরকার এবং এই একটা কনসেনসাসে আমাদের আসা উচিত। হাই ইন্ডেক্স সাসপিশনের কিছু পয়েন্টস আমাদের এখন থ্রো করা দরকার থ্রো ডিজি অফিস। রোবেদ অবশ্যই এটি ইনিশিয়েটিভ যদি নেন যে উইথ হোয়াট ইন্ডিকেশনস আমরা পেশেন্টকে, দ্য ওয়ান থিং ইউ হ্যাভ পয়েন্টেড আউট ভেরি নাইসলি আমাদের আসলে এই ভাগ বটরা করে সিভিরিটির যে ইন্ডেক্স যা আমরা আলোচনা করছি আসলে সিভিয়ার কিনা এগুলি নিয়েও কিন্তু আমাদের কোয়েরি রয়ে গেছে। পসিবলি দিজ আর মেনি থিংস উই নিড টু টেক কেয়ার অফ দিস থিংস। এটি আমাদের একটা প্রয়োজন যে আমরা সাসপেক্টেড কেসগুলিকে কীভাবে ওভারকাম করব, যেহেতু এই একটা প্রোডাক্ট আমাদের একটা ব্রেক থ্রু আনার একটা সুযোগ করে দিয়েছে, আমাদের এটা দেখার একটা সুযোগ এসছে। আমার অনুরোধ থাকবে এসকেএফের কাছে, আমরা লেটাস মেক এ সিআরএফ, যদি দরকার হয় আমিও অংশগ্রহণ করব সেই সিআরএফ তৈরি করতে। উই নিড টু হ্যাভ এ ফেজ 4 সার্ভে। We need to have a phase 4 survey. It's a brilliant initiative being taken by you, bringing the drugs as early as possible. এই সময়টুকুতে আমরা একটা সময় পাচ্ছি, হয়তো বা ডিসেম্বর মাস, যদি আমাদের কপাল খুব খারাপ হয় সামনের ফেব্রুয়ারি বা মার্চে গিয়ে আমরা আবার এক ধরনের বিপদে পড়ব। এখন খুব ইনফ্রিকুয়েন্ট কেস পাচ্ছি, কিন্তু এটা চলে আসবে। এই সময়টুকু আমরা একটু কাজে লাগাতে চাই, এই সময়টা সিআরএফ দেখতে চাই। সারা দেশব্যাপী আমরা এই সিআরএফের উপরে ভর করে আমাদের ফেজ 4 ট্রায়ালের রিপোর্টিং সিস্টেমটাও দেখতে চাই। যদি ডিজি অফিস একটা ইনিশিয়েটিভ নেয়, উই আর ভেরি এগারলি ওয়েটিং ফর দ্যাট। এ কারণে দেশের এই মানুষগুলির আমাদের কি পরিস্থিতি এটিকে আমরা সবার সামনে তুলে ধরতে পারবো। আশঙ্কা কোভিডে হয়তো বা আমাদের আরও বেশি দিন সাফার করতে হবে। আরও আশঙ্কা করোনা ফ্যামিলি যে অতীত থেকে আমাদের মাঝে আছে, নতুন কোনো কিছু জন্ম দেবে। We have to take care of all of these things as a stakeholder to serve our beloved patients with love. Thank you very much for all of you for your patience here.
Thank you, sir. Now I would like to request Professor Harisulok, sir, Professor of Cardiology, BSMMU. Sir, please.
ধন্যবাদ এসকেএফকে একটা চমৎকার অনুষ্ঠান আমাদেরকে উপহার দেওয়ার জন্য। আরও বেশি ধন্যবাদ জানাতে হচ্ছে প্রফেসর রুবেদ আমিনকে। সে আমার মতো অমনোযোগী ছাত্রকে মাইক্রোবায়োলজি ভাইরোলজি সুন্দরভাবে গিলে দিয়েছে। ধন্যবাদ আপনাদের সবাইকে এখানে হাজির থাকার জন্য, এবং ধন্যবাদ জানাচ্ছি এসকেএফকে আবার তার মনোভির বের করার জন্য, এবং এই কারণে যে এখন মাছটা খুলতে পেরেছে এই সাহসে, এবং আমি আশাবাদী যে আমরা হয়তো বা মার্চে এরকম কোনো বড় আঘাত পাবো না। প্রফেসর নজরুল ইসলাম আমার ছোট ভাই, ইনি নিজে করোনায় আহত হয়ে আছেন, ফলে উনি সারাক্ষণ শুধু বিপদ দেখছেন, এবং আমি অত্যন্ত আশাবাদী আমরা মনোভির নিয়ে অনেক দূরে এগিয়ে যেতে পারবো, আর ভয় নয়। এবং আমি মনে করি এটা ভালো বলেছেন প্রফেসর নজরুল ইসলাম যে এসকেএফের এগিয়ে আসা উচিত আরও নতুন নতুন মলিকুলের প্রতি। তাহলে আমরা হয়তো তার হাত দিয়ে দেখবো অ্যান্টি ক্যান্সার ড্রাগ, আমরা তার হাত দিয়ে দেখব নতুন নতুন হার্টের ওষুধ। আমি কার্ডিওলজিস্ট, চরম এক ধরনের হতাশার মধ্যে আমরা দিন কাটিয়েছি গত বছরটা বা তার আগে। আমরা দেখেছি করোনা রোগী এসে প্রথমে যে জিনিসটা রেসপিরেটরি মেডিসিন প্রফেসর রফিকুল ইসলাম ইজ দেয়ার, তাই ওনারা কল পাঠাতেন কি হাই বিএনপি 30,000 40,000 প্রথম কেস, দ্বিতীয় কেস সিভিয়ার প্রলিফেরেশন, চতুর্থ কেস বা থার্ড কেসে পেতাম এমআই এরিথমিয়া। তো এই যে বিষয়গুলি দ্বারা তো হার্ট ফেলিয়ারের প্যারামিটার বেড়ে যাচ্ছে, এমআইয়ের প্যারামিটার বেড়ে যাচ্ছে, সবই কিন্তু সেকেন্ডারিটি কোভিড। এখন আমি বসে বসে ভাবছিলাম যে তাহলে কি যে বিষয়টা দাঁড়াচ্ছে যে মায়োকার্ডাইটিস, অ্যাঞ্জাইনা সবগুলি যেগুলো হচ্ছে, এই ক্ষেত্রে যেহেতু কোভিডের সিকোয়েন্স হিসেবে এগুলো হচ্ছে, তাহলে সাসেপ্টেবল কেসে যদি দেওয়া হয় তাহলে নিঃসন্দেহে আশঙ্কা করতে পারি যে কমে আসবে আমাদের সেই সব হার্ট ফেলিয়ারের প্যারামিটারগুলি, এবং এমআইয়ের প্যারামিটার কমে আসবে। তাহলে অনেকগুলো মৃত্যু, অনেকগুলো সাফারেন্স থেকে হয়তো রোগীদেরকে আমরা বাঁচাতে পারবো। ধন্যবাদ এসকেএফকে আবার এই সুন্দর আয়োজনের জন্য।
ধন্যবাদ স্যার। আমাদের আজকের শেষ বক্তা, আমি বলবো কথার জাদুকর, আমাদের সবার পরিচিত, সবার শ্রদ্ধেয়, আমরা এখানে যারা বসে আছি সর্বজন শ্রদ্ধেয়, আমাদের মেডিসিন সাবজেক্টের পথিকৃৎ, ঢাকা মেডিকেল কলেজের প্রাক্তন প্রিন্সিপাল, বর্তমানে পপুলার মেডিকেল কলেজের বর্তমান প্রিন্সিপাল, বাংলাদেশ সোসাইটি অফ মেডিসিনের এক্স প্রেসিডেন্ট। আসলে স্যারের ব্যাপারে আমার মনে হয় আমি বলতে গেলে আরও আধা ঘন্টা আমি বলতে পারবো, তো আমি আর কথা না বাড়িয়ে সোজা স্যারকে আমন্ত্রণ জানাচ্ছি, প্রফেসর খান আবুল কালাম আজাদ স্যার।
Assalamu alaikum. Good afternoon. Well, as a physician, I am passing through some moments where I feel that I am satisfied. It's very difficult when we found that it's very difficult to fight against COVID-19. A few months back, when remdesivir was there, another program was there in another hotel, and SK+F was pioneer, and I attended that meeting. I talked on some hopes, aspirations, my dreams, expectations, and what was going on at the time. We found nothing at the time. We had no drug, we had no vaccine at the time. A nightmare of uncertainty was there. And at that time, we found remdesivir. Well, remdesivir, the way it works, you know all about that. And also, it was reiterated, re-emphasized by Professor Robed Amin, that, well, the variable working capability of remdesivir made it uncertain to be used regularly in patients with active coronavirus infections. And also, the route of drug is also important. But still, it gave us much pleasure that we got something at the time. But we are waiting that whether a vaccine will be there, whether a very easy way of prescribing a drug for the people having this infection. So it is there ultimately. There are so many questions. There will be so many new expectations based on this treatment, that MOLNUPIRAVIR, whether suppose a patient is there in the house, he is PCR positive, and there are evidences of mild infection of all the members of the family, and they are not ready to go for the PCR test. So that is one way when we can consider, when a disease is pandemic, so we can consider that the whole family, as we saw in the month of May, June, July in Bangladesh. So it happened like that. We treated thousands of patients of COVID infection, and we found that the whole members of the family, they are infected with your COVID infection. So that was a time, that was a really difficult time for us. And also, a person like me, and what I believe that everybody sitting here, all are vaccinated. So if I find that some symptom is there, and if I become positive again, when I am traveling outside Bangladesh, what I'll do? I'll have a very strong temptation, right? Whether I can take this drug or not. What I feel, that many people will feel like this one. So there will be so many questions, so many answers are to be there, especially the vaccinated group of people. But what I believe, that as because the vaccination program is going on, but many of the people are unvaccinated till date. So definitely it requires some type of your treatment. So for treatment of COVID, antiviral therapy, that is the first introduction. What I believe, as the results are satisfactory, we would get a better result in our population. So this is my first point. The second point is that SK+F, what they have done, when remdesivir was FDA approved, and they immediately collected the material, raw material. They started their plant and they started producing here in Bangladesh. So thousands of people got the injection in hospital. And at time there was crisis. We are not getting the injection. But thanks to Almighty Allah and also to Escape. At the time, that was the drug. After steroid, we were thinking of that. So they could provide us this remdesivir. The second point, again, I like to give thanks to Escape that I came here today to give thanks to Escape because I was dreaming of when I first came to know that one drug is there, that MOLNUPIRAVIR, and I started reading on it several months back. But I was considering whether this can be available in the world market. And the UK approved. And then I was thinking that when I will get it. And after listening to it, some of our students were there. They asked me, sir, whether we can get it. I said, definitely, we have got very active companies here. They are very eager to bring the new products in Bangladesh, and especially in this situation. So it happened with Escape. Very importantly, they could bring the drug immediately at the same time of United Kingdom market. So thanks to Escape Bangladesh for their beautiful role. The third point, another attraction for me to come here, and I was with many people in my room, but I know that listening to Dr. Robed is always wonderful. Whenever I listen to him, in the morning, that in scientific session, or in the evening, in cultural session, both are amazing. And I listen to him, and as because Robed is a speaker, definitely I said, no, I have to go. And definitely you found that the experimental part and the pharmacokinetics, dynamics, all were explained excellently, and definitely it is possible because he is Professor Robed Amin. He is such an adorable person with a very strong personality and with much humor, you know. So for that reason, I listen to him, and really I find much enjoyment whenever I listen to him. And today was no exception because he was like that in what he did in Dhaka Medical College. Because in every week we had several sessions in Dhaka Medical College. In the morning sessions we had your grand rounds, we had case presentation, we had journal meeting, and everywhere whenever we used to talk. And definitely Robed was our pearl, and he is doing the same thing here. Thank you, Robed. I love you. So, you know, when I was the principal of Dhaka Medical College, all my gems were taken away from me, snatched, and one after another was placed in different areas. They're starting from the DG, ADG, admin, whatever I say, this Robed, and everybody was taken from me. And the head, Professor Farooq, he was snatched from me, you know. So all these professors, they were with me in Dhaka Medical College. But I had to say, okay, for the betterment of the nation, Dhaka Medical College is always there for the betterment of nation. So, as because I worked with the people who were really the pioneers, leaders, and they are ready to take any active role for the betterment of the nation. So I know what the meaning of this. So today, Robed will be working for our people, and similarly, I want to say, with much thanks to Escape, all the people of Escape who have taken this endeavor, that they are helping our people. Finally, I like to say the undaunted vanguards of our nation, the doctors, nurses, and healthcare workers, because of their extraordinary sacrifice to save the nation during COVID era, we understood, we saw from the front that what the doctors did. So definitely, will remember the sacrifice of all the healthcare providers and still the doctors who are working on it. I know that in front, all the doctors who are here, they are the COVID fighters. My big salute to them. Inshallah, we are working together as because, the viral will be there for a period of time. This, severe disease may go away, but we have to remember that coronavirus will be there. So any type of mutation can occur at any time. So new variant may come and it can attack our people. So we have to remember that. Finally, I want to say that after AIDS, HIV infection, I did not attend so many seminars. When HIV came in Bangladesh, and I had to write so many editorials in paper, publication, I had to go for many speeches on HIV infection. And afterwards, to remind you that when Nipah encephalitis was there in Bangladesh, I was the first consultant to get the patients from Meherpur long 20 years back in 2001, Nipah encephalitis. And one of my intern doctors is here. And at the time, I was associate professor in Dhaka Medical College. And 5 patients from the same family, they came with Nipah encephalitis. At night, when I went to the round, I found that 5 patients from the same family from Meherpur, they are unconscious with high fever, and so many other features were there. And then we didn't know the cause. I was thinking of your Japanese encephalitis at the time because I didn't know about Nipah encephalitis. In the morning when I went to the office at 8 a.m., I asked first that I want to see the patient. But to my dismay, I found that all became dead after half an hour. So, it was a very difficult time, and you know about the Nipah encephalitis in Bangladesh. And again, we have seen so many deaths due to COVID infection. We saw the people requiring oxygen, requiring ICU support for COVID infection. Very uncertain life of a person and wearing this mask. It's very difficult to talk, not taking any water, nothing being eaten by me. So passing for how many days? After 2 years, I am putting on a tie. I just used to live a very simple life within this period. So I hope that everybody present here are very happy today that we have got one drug we can prescribe to our patients, MOLNUPIRAVIR. So again, this afternoon became really colorful as we got something. And after this meeting, when we'll go home, we'll say that, well, some protective mechanism is there in our hand by which we can fight against this COVID virus. Thank you very much. Assalamu alaikum.
Thank you, sir, for your excellent speech. So this is the end of my story today. Thank you. Thank you for your patience hearing.
Thank you, sir, for your wonderfully interactive session. Respected doctors, we'll be heading towards the vote of thanks session now. After the vote of thanks, we will have our lunch and souvenir distribution session. Now, I would like to request the Executive Director, Marketing and Sales of SK Pharmaceuticals Limited, Dr. Mohammad Mujahidul Islam, to give the vote of thanks.
Respected doctors, today's respected keynote speaker, distinguished guests, Assalamu alaikum and a very good afternoon. Firstly, I would like to thank our today's keynote speaker, Professor Dr. Rubaid Amin, for sharing his invaluable insights on Molnupiravir, the first oral anti-COVID drug to fight COVID-19. I'm sure the way our teachers, our senior doctors have already shared that this presentation has immensely assisted all of us in clearly understanding this new treatment approach against COVID-19. To all the teachers, all the respected doctors who have joined with us here today, we are deeply gratified with your gracious presence and our sincere thanks for making this afternoon highly interactive and lively. On behalf of SK+F, I would like to also thank all the participating doctors who have joined virtually from all the 64 districts of the country and also from many countries outside of Bangladesh. To my respected teachers and distinguished doctors, I have many messages in my phone I'm getting from different parts of the world, doctors from Cambodia, Myanmar, Nepal, Sri Lanka, Afghanistan, Uruguay, Uganda, Kenya, many other countries. I'm receiving the messages. And thank you once again to our keynote speaker for this excellent presentation. I am so proud today as a Bangladeshi that just after 4 days of UK MHRA approval, one presentation is being given by one of the leading professors from Bangladesh, which has not only helped the country, I believe it has increased the image of our country across the world. I remember when COVID-19 was declared as a health emergency of international concern. At SK+F, we had an emergency meeting, and there was a clear-cut direction from the management to consider this COVID pandemic as a war. There was one mission only, that we would try and give our best to come and bring any product that will be launched, that will come up to fight against COVID-19. And from SK+F, our pledge was that we will do to the best of our abilities, and ultimately defying all the challenges. Today, I'm very happy that, as all the teachers has mentioned, that only 4 days after UK MHRA approval, we have launched our Molnupiravir brand, Monuvir, as the first generic in the world, which has been manufactured in the plant, and that very plant is approved by UK MHRA, EU GMP, and TGA Australia. And SK+F is already supplying our Monuvir capsule to major hospitals, institutions, and drugstores across the country, and ready to serve the patients as per doctor's prescription. Even to inform all of you that to support the COVID patients outside of Bangladesh, SK+F has already started to cross beyond the boundary. Export of Monuvir has already begun to countries in Southeast Asia and Africa, and inshallah, we are counting for many more. Here, I would like to recall that last year, the same approach was taken. My respected teacher, Kanabul Kalam Azad sir, has already mentioned that same approach was taken in case of Remdesivir, the world's first generic Remdesivir, when it was launched by SK+F just 7 days after UK MHRA approval. And today, I'm very happy to share that even after serving the patients of Bangladesh, our Bangladeshi Remdesivir, SK+F's Remdesivir, has served the COVID patients of 43 countries across the world. And today, you'll be happy to know that even today, thousands of doctors are digitally joining and attending this scientific seminar during this pandemic. নজরুল ইসলাম স্যার বলেছেন যে আমরা অনেকেই সাফার করেছি, আমাদের হারিস স্যার আসার কথা শুনিয়েছেন, আমি নিজেও কোভিড সাফার করেছি। Beloved younger brother Rahan Rabbani is also with us here today. এই সব হতাশা বা এই সব ডিস্ট্রেসের মধ্যেও একটা জিনিস হয়েছে, that a digital revolution has happened. And when physical scientific meetings was not possible, SK+F continued to disseminate scientific knowledge via digital platform with the active participation of senior doctors. And finally, as my teacher, Kanabul Kalam Azad sir, has mentioned, that I want to pay, I will echo with him, I want to pay my sincere gratitude to all the respected doctors, nurses, and all the caregivers who are selflessly providing care to fight the battle against coronavirus outbreak. We pay our deepest gratitude to the doctors, our brave souls who have lost their lives to fight COVID-19. Despite this extraordinary crisis to coronavirus has brought forth, I want to reiterate our unwavering commitment to keep supporting our doctors, patients, and medical communities, and stay true to our belief for a healthier tomorrow. I firmly believe SKF's association with the doctors and the medical fraternity will go a long way in the coming days to make a positive impact in the healthcare sector of Bangladesh and across the world. Once again, our sincere thanks to all the respected doctors who have joined with us today. Thank you very much.